welcome to this class. I am Mobani Kestin, and I'll be taking you on the subject chemistry. The theme of our class is the chemistry of life, and our topic is carbohydrates, part 2. At the end of this lesson, you should be able to group sugars as reducing agents and non-reducing agents. You should be able to explain the hydrolysis of starch and sucrose. And lastly, you should be able to state some uses of sugars. Now, sugars can be grouped into reducing sugars and non-reducing sugars. Now, before we go into what reducing and non-reducing sugars are, it's important for us to know that sugars exist either in the open ring structure or in a cyclic ring form or closed ring structure. So this is to say that all sugars can either have an open chain form or they can have a closed chain form. Some sugars easily exist in these two ways, while some other sugars cannot exist in the open chain form. They are stuck in a closed chain form. Now, in the open chain form, there is something we call carbonyl carbon. And for you to be able to understand reducing sugars, it's important you should be able to identify what carbonyl carbons are. Now, in the open chain form, the carbonyl carbon is a carbon atom that contains the C to O bond. So, here is an image of a sugar in an open chain form, and we say that the carbonyl carbon, to identify it, it is that carbon that is attached to the oxygen of the unit. So, our carbonyl carbon will be this first carbon because it is attached to oxygen. All right? So this is how we identify carbonyl carbon in an open chain structure. Now, in the closed ring structure, the carbonyl carbon is attached to the oxygen of the ring and an OH group. Okay? So there is an oxygen on the ring and there is another OH group attached to a carbonyl carbon. So here we have our carbonyl carbon. So this is how we identify carbonyl carbons in an open chain structure and in a closed ring structure. Now, what are reducing sugars? Now, before we talk about reducing sugar, it's important for us to understand the concept of reducing agents and oxidizing agents. Say you have two substances, A and B, reacting or taking part in a chemical reaction. One of them is a reducing agent, and the other one is an oxidizing agent. Now, what happens is that reducing agents, they are agents of reduction, right? So what they do is that they cause a reduction of whichever substance they are in combination with. Okay? So that is why they are called reducing agents. They reduce the substance that they are acting or reacting with. Now, this substance being reduced is the oxidizing agent. So it means that an oxidizing agent in a reaction is ultimately reduced. Now, an oxidizing agent, it means that it's an agent of oxidation. So it is causing the substance it is reacting with to be oxidized. And this substance it is reacting with is a reducing agent. So it ultimately tells us that reducing agents in every reaction are oxidized. So this is the concept behind what we call redox reactions, with RED meaning reduction and OX standing for oxidation. So it's important we know this because in the course of the lesson, we'll be talking so much about reducing agents and how they are oxidized so that you don't get confused when we say that reducing agents are oxidized and oxidizing agents are reduced. Now, a reducing sugar is any sugar that is capable of acting as a reducing agent because, okay, there are features that make a sugar reducing agent. So, they can act as reducing agents because they contain a free aldehyde group or a free ketone group, and they can exist in an open chain structure. Remember earlier we said some sugars can exist in the open chain structure and in a cyclic structure. So sugars that can act as reducing sugars have to be able to exist in an open chain structure. So this goes to tell us that there are some sugars that cannot exist in an open chain structure. So such sugars are not reducing sugars. All right? And they must contain a free aldehyde or ketone group. So it's important for us to note that reducing sugars exist in the open ring structure and they contain aldehyde or ketone group. Now, all monosaccharides have these qualities. So they are all reducing sugars. They can exist in the open ring structure and they usually contain the aldehyde group or the ketone group. Some disaccharides such as maltose and lactose are equally reducing sugars because they have these qualities as well, as we'll see later on in our class. Now, reducing sugars are so because, remember what we explained earlier, because they cause other substances to be reduced. Whatever substance they are in a reaction with, they cause those substances to be reduced while being oxidized themselves. We said reducing agents, okay, will cause other substances to be reduced while they themselves will be oxidized. So, reducing sugars have this quality, and so that is why they are reducing sugars. These substances being reduced are oxidizing agents, but in the case of reducing sugars, they are weak oxidizing agents. And weak oxidizing agents are usually oxidizing agents that oxidize aldehydes, but not alcohols in basic aqueous solutions. So, example of these substances or weak oxidizing agents that reducing sugars reduce are Tolune's reagent, Fehling solution, and Benedict's solution. Now we'll be looking at monosaccharides as reducing agents. Monosaccharides are divided into two groups, those that are called aldoses and those called ketoses. We've been mentioning that there are monosaccharides that have the aldehyde group. These monosaccharides are the ones we call aldoses, while those ones that contain the ketone group are called ketoses. So, this is what we call the aldehyde group. We write it out in this way, okay? So, these are aldehyde sugars, example, glucose and galactose. Ketoses, an example of a monosaccharide that is a ketose is fructose, okay? You can see that it has this functional group, all right? So we can have sugars existing as aldoses or as ketoses. Now, monosaccharides with aldehyde groups easily act as reducing sugars. They are naturally reducing sugars. But those ones with the ketone group need to first be converted to form an aldehyde group in solution before they can act as what? Reducing sugars. So, these sugars are converted in solution through a process called tautomerism. We'll be looking at the meaning of tautomerism in another of our lessons. So, example of monosaccharides that act as reducing sugars, as we said, all monosaccharides are reducing sugars. So, some of them have galactose, glucose, glyceraldehyde, fructose, ribose, and cellulose. You can go online and check out other types of monosaccharides, okay? Now, we'll be looking at how these monosaccharides work to reduce these oxidizing agents, the weak oxidizing agents. So, monosaccharides as reducing agents, right? Looking at aldoses, remember we said the ones with the aldehyde group are called the aldoses. So, how do they react to reduce weak oxidizing agents? So, we'll be looking at their reaction with Benedict's or Fehling solution and reaction with Tolune's reagent. Now, when aldoses, right, react with these reagents, which can be Benedict's or Fehling solution, are usually solutions of copper sulfate, okay? Either of them, whether Benedict or Fehling solution, would give this same reaction, all right? So, this solution is deep blue in color, but on reacting with an aldose, it will be reduced to a brick red copper oxide, okay? So, that is how reducing sugar can reduce Benedict's or Fehling solution. For Tolune's reagent, all right, an aldose can also react with this weak oxidizing agent, all right, to reduce it to ordinary silver. Now for ketosis, okay, the monosaccharides with the ketone group, okay, they are also reducing agents. And when they react with Benedict's or Fehling solution, they also cause a reduction of this solution to copper oxide. And when they react with Tolune reagents, they also cause a reduction to silver mirror. Disaccharides also, we mentioned that some disaccharides are reducing agents. So this is to say that it's not all that are reducing agents. So what makes some disaccharides reducing agents and others not to be reducing agents? Disaccharides that are reducing agents are so because they have only one of their anomeric carbons involved in what we call a glycosidic bond. Now, this is a disaccharide, okay? From our previous class, we learned that disaccharides are made up of 2 sugar units or 2 monosaccharides, okay? So this is a disaccharide. Now, looking at this disaccharide, you can see that in this linkage, the 2 carbon atoms have been used up to form what we call a glycosidic bond, all right? So at this end, reduction cannot occur. But there is a free end here. And because of this free end, such disaccharide can actually act as what? A reducing sugar. Because this gives it the ability to form an open or exist in an open chain form. Okay? So, because it can do that, and it contains this group, aldehyde or ketone group, right? It can actually serve as a reducing sugar. So just one of their anomeric carbons are involved in glycosidic bond. This is what we call a glycosidic bond. So with the other having an open chain form with an aldehyde group. Now below is a table. We have here a table showing reactions of disaccharides with some of these oxidizing agents. Now, sucrose does not form open chain. Because of that, with Benedict's solution, remember we said that reducing sugars must be able to convert to an open chain form. So because sucrose cannot do that, it cannot act as a reducing sugar. So with Benedict's solution, it will have no reaction. With Fehling's solution, no reaction. Tolune's reagent, no reaction. But maltose and lactose are disaccharides that are reducing sugars because they can't convert to an open chain form. So they can act as reducing sugars as well. So when they react with Benedict's solution, they form brick red solid. With Fehling's solution, they also form brick red solid. With Tolune's reagent, they form silver mirror. So in conclusion, Sucrose is a non-reducing sugar, while maltose and lactose are reducing sugars. So now, I hope you'll be able to differentiate between reducing sugars and non-reducing sugars. You'll be able to tell the key features that make a sugar reducing sugar and make another one a non-reducing sugar. Now, let's look at the key things that make a sugar to be a non-reducing sugar. A non-reducing sugar is one that does not contain a free aldehyde or ketone group. It therefore cannot act as a reducing agent, and also it cannot be oxidized by weak oxidizing agents, as we saw from the table. Examples of these disaccharides are sucrose and trehalose. Okay, they do not contain free aldehyde and ketone groups. Looking at this image, you see that here we have a reducing sugar and another one that is a non-reducing sugar. Now, at this end, this is where reduction occurs. You can see there is a free aldehyde group there that will enable this sugar to act as a reducing agent. But here, at this end of sucrose sugar, it does not have any free aldehyde or ketone group. Therefore, both sides are non-reducing. It cannot act as a reducing sugar. Note that non-reducing sugars are sugars in which the anomeric carbon of the two sugar units are linked together in what we call a glycosidic bond or glycosidic linkage. So we have the two anomeric carbons coming together here to form this glycosidic bond. And because of this bond, they cannot convert to an open chain form. Now, remember from our previous class in the part 1 of carbohydrates, we mentioned that monosaccharides, okay, undergo condensation reaction to form disaccharides, while disaccharides, on the other hand, undergo hydrolysis reaction to form back their monomers. So we'll be looking at hydrolysis of some sugars. And before we go into that, we'll look at what hydrolysis itself is. It's a reaction in which a substance reacts with water molecules to break apart. Okay? So it's like having something solid. Imagine a very solid substance that you have to add water to soften so that you'll be able to have it in fragments. So that is what hydrolysis is about. There is a huge molecule here, and then water is added to it to break it apart. So that is basically what hydrolysis is. Now, sugars undergo hydrolysis to yield back their building blocks. Sugars, whether monosaccharides, disaccharides, polysaccharides, all undergo what's called hydrolysis. And they do this in order for them to be broken down into their monomers or building blocks. Remember from our previous class, we also said that monosaccharides are the building blocks of carbohydrates. So it means that these sugars are being broken down into their individual monosaccharides that formed them. So here is a diagram showing us both reactions, okay? Monosaccharide plus monosaccharide in a condensation reaction will give this disaccharide. Now, this disaccharide, when it wants to form back the monomers or monosaccharides, it does so through this hydrolysis reaction. Addition of what? Water. Condensation had to do with removal of water, okay? While hydrolysis has to do with the addition of water molecules. So we'll be looking at the hydrolysis of sucrose. Okay, sucrose is a disaccharide made up of fructose and glucose, and it can undergo hydrolysis and be broken down into these initial monomers. So when sucrose reacts with water in the presence of an enzyme sucrase, it gives back glucose and fructose. Now, what actually happens is, in the hydrolysis of sucrose, the water molecule present actually reacts, and what it does is that it helps to break down the acetal bond of the disaccharide. When this happens, the hydrogen atom, okay, from the water molecule will now be attached to the fructose, while the OH, water, will be broken down into the ions. So the hydrogen of water will be attached to the fructose, while the OH of the same water molecule will be attached to the glucose. So that is what this water molecule actually does. It donates its ions to these individual sugar units to form them back. And enzyme sucrase, of course, we say that sucrase is an enzyme that is added to this reaction to facilitate it, to quicken it. So this is one way that glucose can be prepared, because as you can see, it's giving back glucose and fructose. So if we want to prepare glucose, we can use the hydrolysis of sucrose to do that. Now I'll be looking at the hydrolysis of starch. Starch is a polysaccharide. It is made up of several units, thousands of glucose units, and it can undergo hydrolysis to give back the building blocks. Starch is made up of several glucose units, all right? So we can achieve starch hydrolysis, one, by acid hydrolysis of starch and enzyme hydrolysis of starch. So quickly, let's look at what acid hydrolysis means. Starch can be hydrolyzed by adding dilute acids, okay, which will yield some intermediates such as dextrin, disaccharides, and glucose. Now, further hydrolysis of these intermediates would finally yield the monomers, which are several glucose molecules. Enzyme hydrolysis of starch. Now, some enzymes are used to catalyze the hydrolysis of starch. They are amylase and amyloglucosidase. Amylase is present in malt and even in saliva. That is why when you eat starchy food, that enzyme is present in saliva to help break down that starchy food you've taken into glucose. So what happens is the enzyme amylase first acts on the starchy substance, all right, to yield these intermediates, a short polymer called dextrin, okay, disaccharides, maltose, and glucose. Remember that maltose is a disaccharide that is formed from two glucose molecules. So these are the intermediates that are given off when the enzyme amylase reacts or hydrolyzes starch. All right. Now this enzyme is like a bulldozer, okay? It comes in to actually act heavily on the starch molecule, breaking down the bonds. All right. Now the second enzyme, which is amyloglucosidase, will further catalyze these intermediates, okay, to yield several glucose units. And I would liken this second enzyme to what I call a sledgehammer, okay? It's not as strong as a bulldozer, but it's also there to help break down those intermediates into their several glucose units. Now we'll be looking at some uses of sugar. At this point, you know that when we say sugar, we're not just talking about the cube sugar you have in your house that you add to your tea. You know that we are talking about carbohydrates. You know that we're talking about different types such as monosaccharides, disaccharides. You know that we're talking about things like starch, cellulose, okay, glucose, fructose, and others. So we'll be looking at some uses of sugars. First one we have here is starch. Starch is used mainly as food. It is found in foods such as our grains and tubers, okay? Two, it is used in the food industry to produce ethanol and glucose. There are several other industries that starch is also used, okay? So you can find that out. Starch is used as a stiffening agent in textile industries and in laundry. Another sugar to talk about is cellulose. Cellulose is a raw material that is used in the manufacturing of a lot of items such as paper, cellophane, and rayon. 2. The human body cannot digest cellulose, all right, but it acts as a roughage in our diet to improve the movement of the digestive tract. Glucose. Glucose serves as an intermediate source of energy to sick persons and to sportsmen. That is why sometimes when you see these athletes, especially runners, okay, you see them take glucose as they are running. It's a source of energy. 2, it is used in the production of jam and sweets. Sucrose. Sucrose is used to produce ethanol through the process of fermentation. It is also added in food, beverages, and hot drinks to sweeten them. So it serves as a sweetener. Sucrose is also used in home baking to add sweetness and texture to cooked products. It's also used as a preservative to prevent perishable foods from spoiling, as used in candied fruits, jams, and marmalades. Then the last sugar to look at here on our board is fructose, which is also called fruit sugar, and it is used in wine making as it is converted into alcohol by fermentation. So we'll just take a quick summary of what we have learned today. We said that a reducing sugar is any sugar that is capable of acting as a reducing agent because it has a free aldehyde or ketone group and can exist in an open chain structure. Okay? Then we said a non-reducing sugar is the opposite because it's a sugar that does not contain free aldehyde or ketone groups, okay, therefore cannot act as a reducing agent or be oxidized by a weak oxidizing agent. Then we looked at the hydrolysis of sucrose, and we said it yields glucose and fructose, while the hydrolysis of starch yields several glucose units. Okay? With this, we have come to the end of our class. We'll take a few questions. Now, which of these sugars is a non-reducing agent? A, sucrose, B, fructose, C, galactose. Well, our answer is A. Remember we said all monosaccharides are reducing sugars, and these are monosaccharides. Sucrose is a disaccharide that is not a reducing sugar. Another question. The hydrolysis reaction of sucrose yield A, glucose and galactose, B, maltose and fructose, C, glucose and fructose. Well, our answer is C. From our lesson, we learned that sucrose is hydrolyzed to give back its monomers, which are glucose and fructose. With this, we have come to the end of our lesson. Thank you for staying with me.
If you enjoyed this video, hit the like button, drop a comment, and subscribe to our channel. You can also turn on notifications to stay updated on new videos on this channel. This Brain Friend video was brought to you by Synforest. For more of these amazing e-learning videos, get your copy of Brain Friend. With more than 1,000 e-learning videos, over 74,000 test items in more than 40 subjects, a career counseling guide, and many other amazing features, BrainFriend remains your foremost e-learning and exam preparatory software. BrainFriend. Learn better, make excellent grades.